It's difficult to imagine a world without flowers. They have intrigued and inspired us for generations. The ancient Egyptians had a very special relationship with flowers, including species such as the blue lotus, said to be the original container for various deities. Flowers have also been found associated with the afterlife. Human burials over 14,000 years old show that people were bringing flowers to grave sites. Flowering plants, the angiosperms, have had an important role to play beyond the aesthetic though. In providing fruits and cereal crops like rice, wheat and corn, the evolution of which we shall investigate later, they have helped to drive human evolution and civilization too. But our association with flowering plants is just a tiny part of the impact that the angiosperms have had on the planet. They are a remarkable part of the biosphere, especially as, on a geological timescale, they are a relatively recent arrival. So let's ask in this episode, what was Garden Earth like before flowers? Where and when did flowers evolve? Let's consider flower power and how did the angiosperms dominate? And also look at angiosperms and animals and see if they were partners together in a great floral takeover. The first evidence of plants growing on the land comes from the Silurian period, about 433 million years ago, with fossils of simple plants like Cooksonia living on waterlogged floodplains. Fossils interpreted as the reproductive spores from these simple plants have been reported from the Ordovician period about 470 million years ago, but these finds are still somewhat controversial. By the time we get to the Devonian, plants are invading drier landscapes, and by the late Devonian there is an incredible innovation, the development of the seed. This, along with other important developments such as leaves, wood and roots, true roots in fact, as opposed to rhizoids, the filamentous hairs you might find in plants such as mosses and liverworts, these developments would finally allow plants to break ties with the water's edge and spread even further into the centres of the continents, with potential negative impacts on the biosphere that we've explored in another episode in this series. Various seed ferns, the Pteridospermatophyta, would flourish throughout the rest of the late Paleozoic, and the vast forests of the Upper Carboniferous, the Pennsylvanian, are responsible for the coal in the Northern Hemisphere that would ultimately power the Industrial Revolution. Seed ferns would flourish into the Triassic and even continue into the Cenozoic, but it would be one particular lineage, the gymnosperms like conifers and cycads, and an array of ginkgo-like plants, benititails and genetophytes, that would really start to advance across the Mesozoic world. Gymnosperm means naked seed, describing the unenclosed condition of their seed, unlike the fruit-encased seeds of the angiosperms. The seeds of gymnosperms form in a whole variety of ways, on the surface of scales or leaves, on short stalks in ginkgophytes, in cones as you would find in conifers, in flower-like structures that occur in the extinct benititails, or in other specialised reproductive structures in groups of gymnosperms that no longer have any equivalents in the modern flora. As you can see, this is a diverse group of plants. By the Triassic, the first period of the Mesozoic, gymnospermous plants represent about 60% of flora species and around 80% of Jurassic species. Today, there are more than 10,000 species of gymnosperms. Today, some gymnosperms, like conifers, produce vast amounts of pollen that they shed into the wind with the hope of reaching a female pollen receptor, a strategy not unlike that used by corals during spawning events when clouds of sperm and eggs are released into the oceans. Not all modern gymnosperms are wind-pollinated though. In fact, many are insect-pollinated. Let's have a look at some of the fossil evidence of early plant-insect relationships in the gymnosperms that might have foreshadowed the special relationship between flowering plants and insects that was to follow. Interesting evidence of this ancient relationship has come from the chemical analyses of the fossil mid-Mesozoic probosces of lacewings. The analysis indicated that specimens did not contain iron in their siphon-like food tubes. If iron was present, they might have been bloodsuckers. Instead, pollen was found associated with the head and mouth parts, which has led researchers like Conrad Labandera at the Smithsonian to suggest that they were feeding on pollination drops, a sugary drop of fluid secreted by gymnosperms to trap pollen grains. In this way, these ancient lacewings were acting very much like butterflies, well before true butterflies would evolve. You may remember we met this particular fossil in an earlier lecture, showing that some even possessed large eye spots, probably to scare off predators, the way that owl butterflies do today. Dr. Labandera and colleagues in France and Spain have found additional evidence of an early insect-plant pollination relationship from the Lower Cretaceous, about 110 to 150 million years ago, of the Basque region of Spain. Trapped in amber are small, about 2 millimeter long insects called thrips, or another common name for them is thunderbugs. The amber they found contained 6 female thrips with pollen grains stuck to their abdomens and wings. Now this was not a chance association, as rings of hairs called rings setae on the abdomen and wing margins appear specifically adapted to maximise pollen adhesion. These pollen collection structures are analogous to the branched hairs found on the mouthparts of modern bees used for collecting pollen. In addition, the pollen grains, most likely from a ginkgo phyte, also appear adapted to be carried by the insects. How could such a relationship develop? A clue may come from the modern ginkgo biloba tree. Even though these particular ginkgos are wind pollinated, ginkgos are either male or female. Male trees have cones that produce pollen, and the female trees produce ovules on the ends of stalks. Pollen has to travel from the male tree to the female tree to allow for fertilization. During the early Cretaceous, pollen from the ginkgo phyte was transported by the thrips between the male and the female plants. It is possible that the bugs were collecting pollen to feed their young nymphs that lived in the ovules of the female trees, ovules that would have provided shelter for a small colony of thrips. The thrips would very selectively transport pollen from the male to female plant host. It could be that insects like thrips were therefore pre-adapted to later exploit the flowers of the angiosperm evolution that began to dominate the global flora during the Cretaceous. So, when and where did this revolution occur? Well, I guess we had better introduce the angiosperms before we get ahead of ourselves. Angiosperms are distinct from gymnosperms in producing flowers and fruits that enclose their seeds by 2 layers of tissue. These seeds also contain endosperm, a genetically distinctive starchy substance that provides nutrients for the growing seedling. The vast majority of angiosperms rely on insect pollination, but as we have seen, it appears that insect pollination was ongoing long before the evolution of the angiosperms. But perhaps the most incredible feature of the angiosperms are their flowers. Flowers are a massive investment for angiosperms, but this is outweighed by the advantages they provide. In utilising animals to aid in their fertilisation, they have employed a very efficient and directed pollen distribution and reception system. The flower contains both male stamens producing pollen and the female pistil containing the ovary and long style topped by the pollen-receiving stigma. The whole structure is surrounded by petals. Flowers will often have the male and female components mature at different times to avoid self-fertilisation. When an obliging insect visits a flower, it deposits a clump of pollen on the flower stigma, resulting in a pollen tube that grows down from the grain, down through the pistil and into the ovary. Sperm is then transferred into the ovule and fertilisation occurs, eventually developing into a seed enclosed by a fruit that may also be dispersed by an animal. Studies of primitive flowering plants like the yellow water lily, Nuphar advena, have shown evidence of an ancient genome duplication that likely preceded the evolution of the ancestral flowering plant. Gene duplication occurs when a redundant second copy of genes is produced. Often this second copy has no effect, but they can mutate over a number of generations, developing a new function that might then be expressed by an organism. This may explain why morphological change, especially of the flowers, could have been rapid. This, though, doesn't help us with the particulars of where and when the evolution of this important group of plants occurred. Charles Darwin was particularly perplexed by the fossil record of flowers. Flowers appeared to just suddenly emerge, fully formed, in the Cretaceous period. This was one of two such problematic fossil appearances that gave Darwin a headache. The other was the apparent sudden appearance of complex creatures like trilobites at the base of the Cambrian, often called Darwin's dilemma, that we covered in an earlier episode. In a letter to one of his friends, Joseph Hooker, a renowned botanist and explorer, dated 22nd July 1879, he referred to the flower issue as an abominable mystery. As with Darwin's dilemma, new fossil discoveries would provide context to the evolution of flowers and help clarify Darwin's mystery, but as of yet, not completely, as we shall see. An ongoing question about the early evolution of flowering plants regards the environment of their evolution. Some researchers suggest that flowering plants evolved on land, but others have contemplated a possible aquatic or at least semi-aquatic origin. One way we can consider approaching this question is to examine where some of the most primitive angiosperms, called annetas, are found today. Anneta is an acronym that stands for some of the basal angiosperm lineages. Assessing which plants are the most primitive can be a difficult task, involving an analysis of what are regarded as primitive or derived characteristics, with plants placed into groups based on this assessment. In doing so, you could build a family tree which illustrated the evolution from primitive to more complex plant species. Fortunately though, taxonomists now have a relatively new tool that provides more of a definitive answer: DNA molecular analysis. Researchers such as Mark Chase at the Royal Botanic Gardens in London and collaborators all around the world have been busy analysing the DNA of angiosperms to provide direct genetic evidence from which to base a comparison of plant species. From this evidence, the flowering plants with the greatest similarity are clustered together in what we call clades. The picture that has emerged has required a reassessment of some of the relationships we assumed before this technique was available. For example, we now know that a rose is closely related to the squash. And consider the beautiful lotus flower, obviously closely related to water lilies, right? Well, no. In fact, the lotus is more closely related to the American sycamore and the London plane tree. What modern molecular phylogeny has also revealed is a plant that is at the base of the angiosperm family tree. This plant represents the most primitive flowering plant that still exists on the planet today, and it is a plant called Ambarella. Ambarella is the only remaining species of the family Ambarellaceae. To find it, you would have to travel to the island of Grande Terre of New Caledonia in the South Pacific. Ambarella is an evergreen, dioecious shrub. That means it produces either male or female flowers, but not at the same time. This is a characteristic more common in gymnosperms than angiosperms, which also hints at its primitive status. It grows in the mountain forests of Grande Terre in two phases. First as a ground-hugging seedling with a creeping root system, and eventually changing into a shrub-like growth when established, deeper root systems are present. Given its basal status, it would seem the tropical upland forest setting of Ambarella would be a good candidate for the environment in which angiosperms evolved. The problem we face here, though, despite its primitive status, is a complete lack of any fossils of a similar plant early in the evolution of the angiosperms. In truth, fossil preservation in such upland environments would be kind of difficult anyway, but not impossible, so perhaps confirmation is out there somewhere. But for now, let's consider the other possible candidate environment, the aquatic environment. For this, we turn to a group of plants that includes water lilies, the Nymphaeales, which do have an early fossil record going back to the early Cretaceous 125 million years ago. Although this lineage doesn't occur at the base of the current family tree of flowering plants, it is pretty close. Surprisingly perhaps, support for an aquatic origin also comes from Ambarella too. Ambarella, like the water lilies, has vestigial gas exchange canals, useful in submerged stems and roots. The aquatic hypothesis is further supported by one of the oldest complete flowering plant fossils thus far discovered. The fossil was recovered from a rock unit in northeastern China, which has become famous for its fossils of feathered dinosaurs, primitive birds, and long proboscis insects of a whole bunch of kinds. The fossil was discovered by Dr. Ji Sun of Jilin University. After studying the fossil, he contacted a colleague at the University of Florida, Dr. David Dilcher, to get his impression of this very unusual plant. The first sample he found consisted of two branches. At the top of the branch were structures that enclosed seeds within carpels, a definitive feature of the angiosperms. But unlike modern flowers, the carpels are spread out along the stem, with possible stamens, the male parts that would contain the pollen, below. This flower, though, lacks petals. It was named Archaeofructus, ancient fruit, and at the time created quite a stir, even gaining the attention of the popular media. The reason for all this fuss was in part due to the proposed age of the fossil. Based on other fossils found in the area, it was suggested that Archaeofructus was growing around 144 million years ago, which would easily place it at or very close to the origin of flowering plants. Could this be the first flower? Drs. Sun and Dilcher suggested that more recognizable angiosperm flowers would evolve from primitive angiosperms like Archaeofructus. Accordingly, the stem that holds the carpels would shrink until they were concentrated close to the male stamens, while leaves were simultaneously modified into petals. The putative late Jurassic age, however, proved problematic, as it conflicted with what we knew about angiosperm fossil pollen. Vast amounts of pollen are produced by plants, and they form a common component of sediments, if unoxidized, deposited in terrestrial settings. Micropaleontologists, and specifically palynologists, had found pollen that could be positively identified as angiosperm dating to around 134 million years ago, 10 million years after the first proposed age of Archaeofructus. This makes the earlier date of Archaeofructus extremely improbable, given the sheer volume of pollen produced by plants. There should not be a 10 million year gap between the first flower and the first pollen. Many of the fossils in this area were preserved when nearby volcanoes covered them in ash, and it would be this ash that would solve the age problem with Archaeofructus. As we have discussed before, volcanic ash is a boon to geochronologists, as it often contains zircon crystals that can be dated using isotopic dating techniques. Once age dated, it was found that Archaeofructus dated to 124.6 million years ago, during the early Cretaceous, after the appearance of the first angiosperm pollen. Although we are still looking for the first fossil flower, Archaeofructus is still an example of a very early angiosperm, and as such might still provide insight into just where flowers first evolved. It stood about 20 centimetres and inhabited the margins of a lake, probably in very damp or even in shallow water conditions, perhaps accounting for its shallow root system. Is this then yet further evidence that flowering plants evolved in an aquatic environment? Well, there are some issues. There are serious concerns regarding the rapid emergence of a terrestrial flowering plant, even accepting assistance from gene duplication from an aquatic ancestor. For example, how could plants that are evolved in water evolve quickly enough to cope with gravity on land? There is no doubt that some flowering plants did evolve in an aquatic environment early in their history, but was this the environment into which the first flowering plants appeared? It is still very much a matter of debate. Regarding the timing of the evolution of plants, based on molecular evidence, it is thought that angiosperms and gymnosperms last share a common ancestor sometime that is very broadly and nebulously called the pre-Cretaceous. Hopefully, new fossil finds will provide us with more insight into the early times of flower evolution. Around 100 million years ago, during the mid-Cretaceous, there was a great radiation in angiosperm diversity, initially noted in the fossil record of angiosperm leaf and pollen remains. By the late Cretaceous, flowering plants started to take over environments that were formerly dominated by ferns, cycads, bennettitales, and other gymnosperms. If you could travel to the late Cretaceous, you would be able to view dinosaurs wandering around plants that you would recognise today, such as beech, maple and magnolia. There is still some uncertainty as to how this replacement occurred. Was it competitive? In this model, the angiosperms, with their short life cycle and rapid growth, were able to muscle in on the gymnosperms and replace them. Or was it non-competitive? We know there was an extinction at the end of the Triassic period that drove many gymnosperm lineages into extinction. Were the angiosperms just occupying empty space ecologically? To completely understand the spread of angiosperms, it would be useful to consider the nature of the world in which they were to rise to dominance. If you consider some of the earliest fossil angiosperms, dated to about 125 million years ago, the planet they inhabited was very different than today. The supercontinent of Pangaea had started to fragment, but there were still large continental blocks, with Gondwana to the south and Laurasia to the north. It is likely that the interiors of these continents would be pretty dry and arid, environments that are particularly favourable to angiosperms, providing them with large areas of the landscape where they could spread and diversify. The innovations that some of the angiosperms evolved included leathery leaves, adapted to prevent water loss, a more efficient water conduction system, and a tough, drought-resistant seed that would stop an embryo from drying out. They also have an ability to propagate and reproduce very quickly, allowing any colonisation to be rapid. Angiosperms are very adaptable and today are found in polar regions, in and along fresh water and seawater, in deserts and numerous habitats in between. Expanding into the dry interiors of continents may also explain why the fossil record is so poor at this time, as arid upland areas are erosive regions, likely susceptible to wildfire events and thus difficult areas for fossils to form. There is another factor though. The mid-Cretaceous was a period of intense warming, probably related to the accelerated fragmentation of Pangaea, an associated generation of oceanic crust and carbon dioxide production. Dr. Huber used microfossils to record this event, as we detailed in another episode of this series. Carbon dioxide may have risen to 4 or 5 times that of today, with temperatures even in the usually deep, cool water of the oceans warming by 15 degrees Celsius. This global warming would have added to intense drought conditions in the centres of continents and further selected for the morphologically flexible angiosperms. There is another hypothesis that has been put forward to explain the explosion of flowers though, a hypothesis put forward by the noted dinosaur paleontologist Bob Bakker in 1986. Dr. Bakker suggests that it may have been changing dinosaur communities that allowed for angiosperms to spread. Around about 144 million years ago, there was a change in the style of herbivory in the dinosaurs, changing from high browsers like brachiosaurs that were cropping leaves from the branches of high conifers to low browsers like ceratopsians. Low browsing would mean that gymnosperm seedlings would have had a reduced opportunity to reach maturity. This would open up the canopy and allow for angiosperms to spread quickly. Due to their rapid life cycle, they were better adapted to reach maturity and thus produce seeds before they were browsed away. Dr. Bakker also proposed that dinosaurs may have been instrumental in spreading flowering plants by eating their fruits and dispersing their seeds. This hypothesis has been largely rejected, however, as no direct relationship has been proven regarding changing dinosaur communities and changes of the angiosperms. However, Dr. Karen Chin of the University of Colorado has documented the presence of seeds in the coprolites of some evidently herbivorous dinosaurs during this time interval. But to close this lecture, let's consider the special relationship that many angiosperms have with animals. The sole purpose of a flower is reproduction, and in the vast number of cases, that means reproduction where a male gamete's pollen are transferred from one plant to another by insects. It is possible that this relationship, and therefore the first flowers, evolved in isolated settings like islands or on an island chain, which might also explain their apparent very sudden appearance in the fossil record. Such isolated settings may have allowed for the development of a specialised relationship between a plant and an animal, for example, a wasp carrying pollen from one plant to another. As we have already noted though, pollination via insects was not a new gig, as various insects had been aiding the pollination of gymnosperms before the widespread appearance of flowering plants. It is likely that some insects were pre-adapted to build this relationship with the angiosperms. The angiosperms, though, would take insect, in fact animal pollination to a whole new level in a classic example of how two major groups of organisms could co-associate over time. Angiosperms, and in particular the flowers, would co-evolve with animals to produce a whole suite of features that would not have existed if it were not for their intimate relationship. To name just a few: scent. Scent would evolve as a guidance mechanism which would allow insects to judge how far away a plant was and just what area of the flower they should zero in on to get a reward such as nectar. Scent, of course, would be a particularly useful strategy if you wanted to attract pollinators like moths in low light conditions. Scent can also act as a timing mechanism, only reaching its maximum potency when the plant is ready to be pollinated. It is thought that scent might have originally evolved from substances that were intended to deter certain animals, but ended up attracting useful pollinators. The actual scent of the flowers can also be keyed to particular types of insect preferences. For example, skunk cabbage and Rafflesia, a flower from Malaysia and Indonesia, are keyed to carrion flies. Another spectacular example from Indonesia is Amorphophallus, the world's largest flower, reaching 10 feet in height. When ready, just like in the previous examples, it emits a powerful stench of rotting flesh to attract cadaver-associated beetles and carrion flies that it dupes into helping pollinate the species. Then there is colour, again used to attract a specific pollinator to specific plants. White, blue and purple flowers are particularly attractive to insects, with reds and oranges being often used to entice birds. In addition, nectar guides on some flowers lead animals to the goodies. Some of these guides are only visible in the ultraviolet area of the spectrum that insects can detect. And of course, there is fruit, generally used to attract larger animals, but also using cues and guides such as colour to let animals know when the fruit is ripe, and of course when the seeds contained in the fruit are ready to be distributed. Seed and fruit dispersal in many ways are the counterpart to pollination in an angiosperm's reproductive cycle. But perhaps some of the most fascinating examples of the closer relationship that exists between plants and insects is seen in mimicry. For example, consider the fly orchid, a plant native to Europe. The flower resembles a flying insect, but more than that, it also releases a scent that mimics the female sex pheromones of certain bees and wasps. The males, attempting to copulate with the flower, get dabbed with pollen. This pollination phenomenon is known as pseudo-copulation and may be the source of great frustration amongst the local insect population. Most of the fossil and phylogenetic evidence indicates that the earliest flowers were small, bowl-shaped, not showy. There were some, though, that would have stood out in the Cretaceous landscape. Some of the first true flowers of the Cretaceous may have resembled something like magnolias. These flowers likely only had a pollen reward for their insect pollinators. The pollinators themselves were likely generalists in nature, such as beetles, short-tongued wasps and flies. By the time we get to the explosion of angiosperms in the mid-Cretaceous, new varieties of flowering plants emerge with new structures, such as nectaries, where nectar is produced, and specialised petals designed for a more specialised relationship with insects. By the late Cretaceous, flowering plants were very diverse. At this time, insects like wasps and flies were also going through radiations, with specific species evolving specific structures to exploit these flowers, such as the long-term bees and flies. Of course, flowers have not stopped evolving. Many of the flowers we see today have been influenced by the selective pressure imposed by humans. Some have been selected by humans for the fruit they produce. But it is possible that many domesticated non-food flowers probably started as weeds in stands of other plants cultivated by humans. It is likely that some of them didn't get weeded out due to their appearance and perhaps were perceived as being beneficial to the crops that were being cultivated in some way. The end result of this radiation of the angiosperms during the Cretaceous would be a world with a radically different flora and companion insect associates to match, a much more colourful world of fruit, flowers and exotic scents. It certainly is a beautiful world, even if it is all down to sex.